Thank you all, and welcome, and thank you for joining us for the webinar from Joanne Donovan from Catabasis. Today we're going to talk about idalacilonexant. I hope I've pronounced that correctly. My name is Pat Furlong. I'm president of Parent Project Muscular Dystrophy, or PPMD. We're going to talk today about an NF-kappa B inhibitor, and if we think about where we are with this, we're all quite familiar with the use of steroids, whether it's weekend steroids, 10 days on or 10 days off, or daily steroids. And we've been used to those for many years, and certainly they are the standard of care. What we see in steroids is the negative effects on linear growth, on bone, on behaviors, and on weight. And for a long time, while we appreciate the effect and the impact of steroids on walking and muscle function, we really would like to get around those negative effects. When we think about what prednisone or ZScore and Plaza do in this case, they really suppress the inflammatory response or inflammation due to muscle degeneration. And if we think about where this starts, it actually starts in the fetus when the fetus has utrophin on board, the protein that we're talking about substituting, but at any rate, that's a fetal protein. When that fetal protein begins to change and transfer into dystrophin, and then recognizing dystrophin is absent because of the mutation causing Duchenne, then inflammation starts in the fetus. When the baby is born and starts to move around, inflammation increases. And so the use of steroids is to really decrease that effect and therefore slow muscle degeneration and preserve function. Getting around those side effects is going to be important, and I think all of us would like to give up those steroids for a drug that would have equal benefit and perhaps much less side effects to contend with. So today we're going to talk about a drug that is in the NF-kappa B pathway, and this is one of the most significant pathways of inflammation in Duchenne muscular dystrophy. So with that, I'm going to turn over the presentation to Joanne Donovan. Joanne is Chief Medical Officer for Catabasis, a Harvard-trained physician who will lead this effort, who is leading the effort, and will lead the webinar today. So Joanne.
Thank you, Pat. First of all, I'd just like to say I'm delighted to be here to share what the new information that we have on idalacilonexant. We have been working on this and working with Parent Project for a long time, and we have very much appreciated the support that they have given us along the way to get to this point where we believe that idalacilonexant is a promising agent for the treatment of Duchenne. Now, idalacilonexant is an oral small molecule that, as Pat says, is designed specifically to inhibit NF-kappa B in order to treat Duchenne muscular dystrophy. Now, Catabasis is a public company, so I do need to show you the next slide, which says that these are forward-looking statements. It's for investors, potentially. So we'll move on to the next slide, which gives a little bit about the background of why we are using idalacilonexant and developing that for Duchenne. Okay. So we know that NF-kappa B is activated in Duchenne following mechanical stress. And in fact, the absence of dystrophin is the fundamental cause of the disease, but it's actually not the only thing that causes the disease process to progress in Duchenne. And it's a combination with mechanical stress that actually activates NF-kappa B, and that activation of NF-kappa B then promotes muscle degeneration. It suppresses muscle regeneration. So for several years, we have been working with experts in the field, Professor Lee Sweeney at CHOP and UF, to understand the effects of NF-kappa B inhibition in the MDX mouse model for Duchenne. And in that model, Dr. Sweeney has shown that inhibiting NF-kappa B with idalacilonexant, or a closely related molecule, then prevents these effects of activation of NF-kappa B by decreasing inflammation, by decreasing fibrosis, by allowing the muscles to regenerate, and ultimately to improve function in the MDX mice over a period of time. So based on this, we've gone forward to develop etasolonexant for boys and young men with Duchenne. And as you well know, the disease is characterized by a very predictable sequence of effects where the boys lose function and they lose certain aspects of mobility. We've been looking in the boys that are 4 to 7 years old. And in those boys, you see changes in the ability to hop, to run, to jump, and that's followed by the loss of the ability to rise from the floor, to climb stairs, and then subsequently followed by the loss of the ability to walk. So while many of the trials that you have been seeing have looked at the 6-minute walk time as a major outcome measure to examine in trials with patients with Duchenne, we are looking at younger boys, and we have been using different outcome measures. We've been using the North Star Ambulatory Assessment, and we've been using measures such as the time to rise from the floor, the 10-meter walk-run, and the time to climb stairs that are all very much focused on the way that Duchenne affects the abilities in this age range from 4 to 7. And ultimately, our hope, as your hope is to be able to delay the loss of mobility and the loss of functions, and to be able to slow that decline. And we have very encouraging data in the trial that shows that etasolonexant has promise in that way. So I'm going to focus on the next slide on the design of the MoveDMD trial. It's complicated, so I will take some time to go through it. We have talked before on a webinar as we progressed through this design. And prior to starting the MoveDMD trial, we had looked at etasolonexant in adults. We had seen no safety signals. We had seen that etasolonexant could inhibit NF-kappa B. And we then went into a study where, for the first time, we looked at it in young boys, age 4 to 7, confirmed Duchenne, and boys that were not yet on corticosteroids, since the average age at which boys start corticosteroids in the United States is around 6. So the first phase 1 part of the study was looking at a period of 1 week where the boys would get one of 3 doses of etasolonexant. And that part of the study took months to complete because we had to look at the data between each dose and decide if it was safe to go to the next dose, and then be able to get all of the regulatory approvals to be able to go into the next part of the study. So there was an off-treatment period. Boys were treated for a week, and then they were off treatment for what turned out to be an average of about 9 months between that and the start of the phase 2 part of the study. Now, at the beginning of the phase 1, we also measured the functional measures in the boys, the time function test, the North Star, and the MRI that was important in the design of the study. We enrolled 17 boys in that part of the study, and 16 of them continued on to the phase 2 part of the study. And we enrolled an additional 15 boys. Those 31 boys were randomized into one of 2 doses of etasolonexant, 67 or 100 milligrams per kilogram per day, or placebo. And we had picked those 2 doses because in the short phase 1 part of the study, they were shown to be safe, and those 2, which were the higher doses that we looked at, were also shown to inhibit genes that respond to NF-kappa B. So that's why we took those into the next part of the study. The next part of the study was 12 weeks placebo-controlled. As you've heard, the FDA does want to see a placebo-controlled from as soon in the development program as possible. And we looked at the beginning and end of that period at the time function tests at MRI, and of course at safety throughout that period. We presented the results of that 12-week period in February of this year, and we had planned to look at MRI as a biomarker endpoint that could give us information in a short period of time about the effects of etasolonexant. And we saw that biomarker moved in the right direction, but it did not reach statistical significance. What we also saw, though, which was encouraging to us, was that there was no safety signal in either dose, and we saw consistently that the higher dose of etasolonexant, the 100 milligrams per kilogram, was better than the placebo. So this was encouraging, but it was a short period of time to look at the functional assessments. Now, all of the boys went on to open label drug after that, either 100 or 67. But when we got the results of the phase 2 part of the study, the Data Safety Monitoring Board and Catabasis looked at the data, and we decided that it would be a good idea to move all of the boys to the higher dose, the 100 milligrams per kilogram, since that was safe in the studies that we've done so far, and it had the best possibility of showing an effect on function, which we were going to be looking at in the open label extension. So in the analysis that we presented last week at World Muscle was looking at the open label extension, where all of the boys know they're on the drug, and the analysis was done when the last of the boys were on drug for up to 24 weeks. And so that whether or not they began drug at the beginning of the phase 2 part or the beginning of the open label part, that counted as the start of the 24-week period. And since many of the boys had been on drug since last summer, many of them were also now at 36 weeks. So here the data we're presenting are with everybody getting to 24 weeks after the start, and many of them getting to 36 weeks. So on the next slide shows you, or 2 slides shows you how we are analyzing the data. And what the design of the study allowed us to do, because it's very complicated in terms of knowing how the boys are changing if we didn't intervene with a medication versus when we are treating them with etasolonexant. And the design of the study allowed us to look at this control period of about 9 months on average before most of the boys got drug, and compare that period with the period when they were on etasolonexant in the open label extension. So for all of these, we have on the vertical axis the speed of the time function test, or the North Star score, and on the y-axis time, with the clear area on the left being the 39 weeks before they started treatment, and the blue line showing the rate of change, which was actually a decline during that period. And the green area on the right is showing what's happening while they're on etasolonexant. And the projected line there is basically what would be continuing to happen if they were not on treatment. And that's probably a conservative estimate because, as you know, the boys often things happen faster as they get older in age, and many of the boys in the study are now up to 7, 8, and even 9 in the study. So on the next slide is the first of the functional measures, the 10-meter walk-run. So they're asked to run or walk as fast as they can for 10 meters, and they do a good job. They do it 3 times. We take the fastest of the 3 measure. And when they were first evaluated, this is the speed on the y-axis. The speed was 0.17 per second, and that corresponds to about almost 6 seconds. At the end of that control period, they averaged a speed of around 0.14, which corresponds to about 7 seconds. So over that period, on average, they took about a second longer, which is about a 14% loss of speed. And, of course, this is not an atypical period. This is something that's happening every 9 months for these boys. And to compare, an unaffected boy would probably take about 3 seconds to complete this task. So what we do see is on etasolonexant, 100 milligrams per kilogram, starting at 0 weeks when they start on etasolonexant, instead of the continued decline, we have stabilization, that they are able to continue to do this at the same rate of speed. On the next slide, we have the 4-stair climb. And there they went from around 4 seconds at the beginning of the control period to about 5 seconds at the end of the control period. And an unaffected boy would be probably about 2 seconds for being able to climb 4 stairs. The 4-stair climb is lost before the ability to walk, and this is moving down more quickly, about 24% over that 39-week control period. And again, we see that the 4-stair climb speed is stabilized over the 36 weeks on 100 milligrams per kilogram etasolonexant. So we're very excited to see the consistency, again, another measure that we're able to see them stabilize over 6 and 9 months. On the final time function test is time to stand. And there they started out with a speed of about 0.23, about 4 seconds, compared to around 2 seconds for an unaffected boy. And they lost they lost speed over the period of 39 weeks. And whereas on etasolonexant, 100 milligrams, they were able to be stabilized. Lastly, with the North Star ambulatory assessment, this is a composite of 17 different functional measures. Many of you probably have seen this happen. And the trained observer scores the boys on each of these 17 measures. So it really does capture hopping and jumping and all of the kind of things 4 to 7-year-old boys do in daily life. The maximum score would be 34, and at the beginning of the control period, the boys had an average score of about 22, but fell to about 16 at the beginning of the etasolonexant period. And then again, there was a clear stabilization over the 36 weeks compared to what would have been expected to a decrease of about 5 points. So we actually have heard anecdotes and seen in the scores of this North Star that some boys were able to run better, they were able to hop better, and that's certainly very, very encouraging for us and for the parents in the study to see. I think while these individual tests are not statistically powered for significance, what we have found, and the experts we've spoken to have found very reassuring, is the consistency of these over the 4 tests. In addition,
Joanne, Joanne, can I just interrupt you for a minute? I think it would be really useful for you to explain. So the North Star is a series of 17 different activities that are measured. Would you explain to them about the scoring and how that's done, and what a good score versus a perhaps a less good score is, and just so they understand how that's measured and how you get to these numbers of 34 and what that might mean in terms of difference.
So thanks for asking. That's a good clarification. So the different measures are range over really an age range from things as simple as standing to running to standing on one foot, standing on the other, hopping on one foot, hopping on the other, jumping with both feet clearing the ground, and going up and down stairs 1 foot or one step at a time with each leg. And the score, if they can do it without any noticeable difference from an unaffected boy, they get a score of 2. If they can't do that at all, it's a score of 0. And if they can do it, but they compensate for it, maybe for getting up off the ground, they're doing the Gowers maneuver. So if they're compensating for it, then they get a score of 1. So you see at the beginning of the etasolonexant period, with a score of 16, they have lost a significant number of these abilities to be performed as an unaffected boy would be. And in fact, they are at a point where they are typically declining about 4 points per year. So the reason that the North Star is a measure that regulatory agencies like is it gives a broad picture of the functions, the abilities of boys in everyday life, the things that parents see every day and are important, the ability to climb a stair, to get up from the floor. And so that's the reason why we think this is an important measure and are very encouraged to see the stabilization in North Star over this period of time.
Thank you. And so an increase from baseline, if you started out at 16 points, meaning that indicating that you needed help with certain of those measures, so scoring a 1 versus a 2. So increasing in score means that that child is increasing in the ability to perform those functions.
Yeah, exactly.
Good. Thank you.
Good. And so we also looked at other measures of muscle health. And one that we look at as a safety measure is different muscle enzymes. The CK creatine kinase is elevated almost 100-fold in boys of this age. And it's a measure of, it is increased because the muscle plasma membrane becomes leaky. And all of the muscle enzymes are increased because of that. These decreased, and they decreased over the first about 12 weeks of treatment, and then stabilized after that. And these differences were statistically significant for all of the 4 muscles, the enzymes that we looked at: the creatine kinase, alanine aminotransferase, aspartate aminotransferase, and lactate dehydrogenase. So these are all enzymes that are known in the blood to be elevated in boys with Duchenne of this age. And we think that the decrease in muscle enzymes is consistent with a positive impact on the muscle and supports that etasolonexant is working and improving the muscle function. We also looked at MRI. And MRI is a non-invasive measure to look in a magnetic field to understand more about the muscle chemistry and physiology. And we looked at a composite measure of the 5 lower leg muscles. So we averaged the signal from the 5 lower leg muscles, and that's called the T2 signal. Measuring this is no easy matter. The boys, it takes about an hour or so, an hour and a half in the MRI scanner. There's no needles involved, there's no sedation, but it certainly was a part of the study that was challenging for some of the boys. So we very much appreciate their efforts and their parents' efforts in doing this, because it did give us very important information. And this is being done in conjunction with the Imaging DMD group, which Parent Project has supported for a number of years, in order to understand using this as a biomarker in studies with boys with Duchenne. And what we see in the blue bar, the control bar, is the average MRT2. This is elevated in boys with Duchenne compared to boys that are unaffected, and it increases year after year. And what we saw, in contrast to the increase that we saw in the control period, after 12 weeks of etasolonexant, 24 and 36, we saw that this was stabilized. It no longer increased over that period of time. And this, again, was statistically significant. So we're able to look with a very objective biomarker and see that there is a positive effect of etasolonexant on the muscle, and supporting that it's being stabilized over time compared to what would happen in the absence of etasolonexant. So we will be continuing to collect data on MRI and sharing with Imaging DMD, because this is an important biomarker, particularly in a phase 2 study like this, where there's a smaller number of boys that are enrolled. So this is very important information for us, and also for the field to be looking at how this changes in drugs that affect the muscle in Duchenne. So safety is incredibly important, because we're looking at any of these therapies as long-term therapies. And we have not seen a safety signal in the MoveDMD trial. We're now looking at over 25 years of patient exposure, so the equivalent of 25 boys for a year. What we saw was that etasolonexant was well tolerated. The most common adverse events, side effects that were seen, were gastrointestinal, and the most common was mild diarrhea. It was usually described as loose stool. The average time that it lasted was a few days, and it usually happened at the beginning of treatment, within the first couple of weeks, and then it seemed to resolve over time. We didn't see any serious adverse events. We didn't see any, the drug was never reduced because, to a lower dose because of the diarrhea or because of the GI side effects. We looked at vital signs carefully in the study, and EKG. And one of the things that you may know is that a rapid heart rate or tachycardia is seen in the boys even at a young age, before they have any of the cardiac complications. So it's the very first thing that is seen, that the heart in any way is noticeable. And what we saw was that the heart rate over the 36 weeks decreased towards the normal heart rate that you would see in boys of this age. We have been looking at the effect on the heart in the animal models, and we have seen positive effects there. So this is something that is encouraging, and we'll certainly be looking at in future studies to understand what it could mean. We didn't see any changes in labs, in chemistry labs, in the kidney function, adrenal function, calcium or phosphate. And one of the reasons for looking at calcium and phosphate is that those are in the blood. The calcium level falls, for example, with steroids. We know that they have a negative impact on bones. So we don't expect that, since etasolonexant is not a steroid, we don't expect it to have that effect, but it's always reassuring to see that the blood tests related to bone have not changed. We also looked at growth, and we are showing here what the percentile for weight, height, and body mass index is. And the boys, so this is comparing this to standard growth curves, to growth curves of unaffected boys. And the weight remains flat. That's what you'd like to see. You don't want to see it increase. And the height also, they continue to grow. If they're staying on the same percentile, it means they're growing as the average boy does. And so their height was increasing, as you would expect, over the period of the trial. And their body mass index, which is the weight and height combined, tended to go down a bit, which was actually towards more normal, towards an average value. So they had very age-appropriate increases or changes in their height and weight, which is reassuring. So to summarize, what we found from the open-label extension is that we saw that the disease progression that we had seen in the control period was decreased in the period when the boys were on etasolonexant 100 milligrams per kilogram. I would note that we are showing the results from the 100 milligram per kilogram because we decided that that was the dose going forward once we had the results last February. We were able to see these improvements for the North Star and for the time function tests that are age appropriate. And we also saw the changes in the MRI and muscle enzymes that give us additional support for the biological effect of etasolonexant. And importantly, there was no safety signal, it was well tolerated, and the growth patterns continued along normal, unaffected curves, which was great. So the reason that we really wanted to see the changes in the functional endpoints is because that's what we would use for a phase 3 trial, which we expect to start the first half of next year. Before I want to tell you more about the phase 3 trial, and we've been able to design that based on the results that we've seen. And we're planning this to be a 12-month randomized double-blind placebo-controlled trial of the higher dose, the 100 milligram per kilogram versus placebo. And after 12 months, all of the boys would continue on active drug in an open-label extension. We have had the opportunity to talk with FDA, and so this design has been informed by that, and so we are at a good stage moving forward in terms of the planning. And we will be looking at 4 to 7-year-old boys, and we'll be looking at the same endpoints we've looked at previously in this MoveDMD study. Again, this will be boys that have not been on steroids for 6 months or are steroid naive. And the endpoints that we're looking at, we want to listen very carefully to what the FDA said last week in AdCom and before. And these endpoints, the North Star and the time function test, are very much in keeping with what they have advised us and what they've been advising in their draft guidance for boys of this age. We are planning to enroll about 125 boys with a 2-to-1 ratio, so 2 boys getting etasolonexant for every 1 boy that would get placebo. And lastly, I just want to touch talked about the other therapies, and we've been working with Sarepta to understand how etasolonexant could work with exon skipping therapies. We know that NF-kappa B activation actually causes a decrease in the production of dystrophin. And the boys in the trial that we just have completed don't have dystrophin. But if you have a dystrophin-focused therapy like eteplirsen, then it could be an additional way that the drug works for etasolinexin to be given along with eteplirsen. And we looked in MDX mice, and this is the familiar picture on the top of this staining for dystrophin. The MDX mice where it says saline do not show dystrophin production. With exon skipping, they do show, you can see the outlines of multiple cells there. And with the combination of exon skipping and EDASA, we saw even more dystrophin produced. So the measurement of this went from about 4 to 7% in mice, which was statistically significant. So we have allowed boys that are 51 skippable in the open label extension. There's a couple boys, and they're starting treatment with the now approved drug, eteplirsen. And we anticipate that boys that are on a stable dose of eteplirsen will also be in our phase 3 study as well.
So thank you, Joanne. Just a question while we're on this subject of potential combinations. So the boys who were in the previous study, I think it was 28 or 30 boys that have continued through the current study that you have just analyzed and presented, will those boys be rolled into the double blind, or are those boys a separate cohort that may be amenable to those that are amenable to an exon 51 skip potentially will have exposure to that or be able to continue on that? So these boys in this first cohort of children that were treated with edalasolonexant, we have to really shorten that quick because it's a mouthful. Will they be rolled over or considered for the phase 3 study, or is that a separate cohort that will continue extension until a decision is made and then have an opportunity to participate in for instance, exon 51 or potentially other drugs as they become approved?
The boys that are currently in the MoveDMD trial are continuing, and we anticipate we've now extended this to, I think, about 2.5 years total of treatment. And if they are eligible for eteplirsen, then they can start on eteplirsen. That's about 13% of the overall population, so it's a minority of the boys. And certainly, if there are other dystrophin-focused therapies that become available, then we will look to make those changes as well.
Good, thank you.
Boys will not go into the phase 3 study because we'll be continuing them on in the open label extension.
Right, and then so just to be clear, so in this phase 3 recruitment that you're going to begin in the first half of 2018, if one of these young boys is on exon 51, the approved drug, that would not be part of the exclusion criteria. Rather, that would be considered steady state, and he could be amenable or be fit into the inclusion criteria in terms of steroid naive or steroid free for 6 months prior to being included in the study.
Exactly. And that's where what we our ultimate objective is to develop etasolinexant for all boys with Duchenne, regardless of mutation type, and potentially also is monotherapy, but in combination with other modalities, and really have it, the goal is to make it a foundational therapy in Duchenne.
Yes, certainly combinations is what we've been thinking about, dreaming about, and driving toward for many years. So this is really a terrific opportunity for boys who are amenable to exon 51 within your study. So thank you for that.
Yes.
A couple questions just as we go along. This was earlier, and one other question about this current and planned trial to open in the first half of 2018. 100 milligrams per kilogram is the dose that you've chosen for the study. Given the safety profile of this drug, have you looked at higher doses, or is this what you've just determined based on preclinical that the 100 mg per kg is the dose recommended for the therapy?
So the boys are taking this in capsules that are about 2/3 of the size of an M&M, and they're all doing a great job with the capsules. We are in the open label extension. We are going to look at a higher dose in the next several months, and because it's a very important question, could you go higher? Could you get more effect, and would it be safe? So we want to answer that question in the near future and understand that as well. We have the drug. There is a limit in terms of how much can be absorbed, so we probably can't double the dose, but we are going to try to look to a slightly higher dose and see if that has an effect. We have in our science group has looked and they have data to support that going to a slightly higher dose might be very helpful as well.
Good, and then you would amend the study and go to a higher dose once you've concluded that, or you would start a small safety study and then amend the protocol. How do you see that being integrated within the planned study, phase 3?
So we are going to take it one step at a time. We'll see what the data shows on these boys and then decide on how to include that in the phase 3 study or potentially in a different study.
Okay, great. Thank you. So a couple questions have come in, Joanne. One is a question around the data. Have you broken it apart in groups based on the length of time people are on different doses and taken a look earlier, or really looked at the collective data over time once you switched everyone to 100 mg per kg?
So because it takes a while for things to get through the IRBs to get approved, the boys, and right now the boys are coming in every 3 months for these assessments. So we don't have a lot of data on the boys that were on 67 and moved to 100. So we will be looking at that going forward, certainly.
Okay, thank you. And another question around statistical difference between baseline and the 36-week time point. Did you see differences at that 36-week? And I think the question is around the fact that in the studies that we've seen come through, that we've been involved in and the data has been shared, what we often see is that we don't see a difference early on, but we see a difference, a cumulative effect over time, and it takes a little bit longer to understand the difference and the degree of difference. So one of the questions is there any statistical difference between baseline and the 36-week time point?
Yeah, those time points are not statistically, those are not different. We did not power the study in that we wouldn't expect to see a difference with 16 boys. To be able to see differences, we need to look at a larger study, and we're estimating that to be about 125 boys. What does give us encouragement is that for all of these measures, we are seeing the stabilization. So does that answer your question? Okay.
Yeah, it does. It was a small study of a small number of boys, and I think that the community is quite used to the fact that we do need bigger numbers to see differences. And so in this next planned study, are you thinking of interim analysis, or are you going to go the full length and then look at the data at the end of the day?
Yeah, you saw that the dashed line and the line of IDASA, that widens, that we saw some effect at 12 weeks, but it has seemed to widen as we went to 24 and 36 weeks. So that's why we are looking at a 12-month study. We'll certainly be doing interim or looks for safety and have a data safety monitoring board to look at that. But because the effect does seem to take time to diverge, we think that the fastest way to do a study to show a difference between placebo is to do a 12-month study.
Right. Yeah, that makes perfect sense. It's just that, we need things yesterday, and so we always are going to ask questions, so I appreciate that. And I guess the other question that has come in is, should you determine that perhaps a higher dose might be a better option, will you go back to the first study and increase the dose that they're all stable on 100? Will you increase them to whatever dose you found has been both safe and potentially more effective? Or have you thought, or you might even, taking it one step at a time, not be there yet? But yes. Just a question.
The boys in the MoveDMD will be the ones that move to the 133. So they won't be on 100 anymore. They will be on 133 as long as everything goes out there.
So you'll move those up and continue with the phase 3 plan study. So if everything goes well, here's a question of when, right? When would you see this drug? Again, this is all based on if this, then that, and it's a calculation that we make in our head every single day as parents. When is the most near-term possibility of an approval in this drug? Should everything go that way?
Right. So we are trying we've looked at the timeline for enrolling the study and then the 12 months for the study and analyzing it. That puts us having results in about 2020. And so that is, we dearly wish that would be sooner, and we know that we will continue to work with you, with Parent Project and other groups to try to move things along as quickly as possible, as we know that the importance and the urgency to the community. That's our current estimate, and we would do everything we can to move it in quicker.
Great, thank you. Now, always, as you know, we have young adults who who have lost the ability to walk, who may, are not on steroids. Some of them have discontinued steroids when they lose ambulation for a variety of reasons. Doctors' decision by the person or the family, for many other reasons. So one of the questions is, have you considered or will you consider a study in the non-ambulatory group of young individuals, as well as you know, Joanne, I don't think we've seen in this world, other than one study, a study of adults over the age of 18. So take those two apart, and the non-ambulatory study, have you considered it? The second would be an adult group, and then the third is just a comment from you about your company's position on compassionate use.
Okay. So we have been spending a lot of time thinking about the best way to do a study in non-ambulatory boys and young men. And we know that there are other companies that are doing studies where we're trying to understand the best way of doing that, and that is something that we do intend to do as part of our development plan, because our goal is to bring the drug for patients with all mutations and really at all ages as well. So that is something that we are planning going forward. And in terms of young men over the age of 21, I think that we have had the opportunity particularly with a lot of the symposia that PPMD has put together, understanding how to look at enrollment in those studies and to really try to not base it on age is important. So we hear that message, and as we design the next study, that's our goal as well.
Great, thank you. We so appreciate that. Sorry.
I was going to say, in terms of compassionate use, that is something that we are not doing compassionate use at this point, but it's something we will continue to evaluate as we move forward.
Thank you, and we'll continue to ask, so I appreciate that. I'm assuming that when you embark on a study using patients who are no longer walking, you'll think about using the pull or the performance of the upper limb. This is being used by any number of studies, and certainly has has really been, as you know, developed by patients. Certainly our Dutch friends, Duchenne Parent Project and PPMD have really initiated the development of the pool with Natalie Gomans and others. So we're assuming that you'd include the pool into such a study.
Mm-hmm. Yes. Yes, we would, and respiratory measures as well.
Right.
And also thinking about cardiac measures as well, which is very important in that age group.
Yes. Totally agree, and the more we're trying to really learn and work with FDA about what is the best measure or what is a reasonable measure or reasonable outcome to use for the heart. As you know, that's not exactly simple, but we're all trying to really dive down and figure out what might be useful and what might be agreed upon to go forward. We talked about the MRI and the T2 images, and it changed from 24 to 36 weeks. Mm-hmm. So the question is, and you mentioned this is an ongoing study, and so I'm assuming that it's ongoing in the MoveDMD trial, and you'll continue to look at the T2s throughout that population. And for the next weeks, is that planned, and is it planned in the phase 3 study?
We are going to continue to work with Imaging DMD to look at MRIs in these boys. We are doing it less frequently than we did at the beginning, so trying to reduce the number of visits and certainly the burden on the families. We are not planning to include it in the phase 3 study, given the larger number, and that the endpoint that the FDA is most interested in is of a clinical endpoint. That being said, getting longer-term data with MRI, with Imaging DMD, I think will be important in providing background information for etasolonexant and also on the disease progression in Duchenne. And so we're planning on continuing that effort in the MoveDMD study.
Thank you. I do think the T2 images are really helpful and certainly supportive and support the functional outcomes as they improve or stabilize. So I think that that's terrific, and I can imagine with the numbers you're recruiting for your phase 3 that it would just become really an overwhelming kind of study, both for you, for the families maybe, as well as for the clinical sites. So we certainly appreciate the continuation of the look at the MRT2s throughout the MoveDMD study. So let's see. You've mentioned some of the results that were not statistically significant but were consistent over time. Have you looked at the nonparametric significance test for this consistency? The first nonparametric test is the RUNS test, though others might be worth considering.
That's a good question. We have looked at both kinds of methods to compare them. The numbers are relatively small, and I think that when you look at the size of the phase 3 studies, not just from us but from others, to power a study adequately does require a much larger number of individuals.
Yeah, I thought maybe you had too few in the earlier study in the MoveDMD study to really look at that very well. So we do appreciate that, but perhaps in the next study you'll be able to better look at that. Mm-hmm. So, Joanne, first of all, I can't thank you enough for this. This has been really informative. We are certainly anxious for you to start. I guess one of my final questions would be, we all know the impact of steroids on bone health, and we see these Z-scores that are really at baseline below 1 or 2 standard deviations below normal. And more recently, we've had a bone health meeting that you attended and talked about the fact that maybe the Z-score is a good method, but perhaps it might be more important to look at the spine films and to look for compression fractures. I guess what I wondered in your phase 3 or even your MoveDMD, are you looking at Z-scores? Will you look at Z-scores? Are you going to look at spine films, or is this really you're looking at functional outcome and that sort of gets over the top in terms of measures for a study?
Those are wonderful questions, Pat, and that is one of the most important morbidities of steroids, and it's something that there's a lot of data in the literature that suggests that inhibiting NF-kappa B could have a positive effect on bone growth without the negative effects. So what we are looking at what the best way, and we've been talking, one of the having had that symposium, the Bone Health Symposium, we've made contacts with a number of folks in the field of bone health in Duchenne. So that is something we've been talking with them about, what are the best measures in younger boys to look at. So, yes, more to stay tuned.
Okay, great. And I think there's always going to be a question of why a placebo and can we use natural history controls. And I think I'll say that, you know, we are all gearing into this with critical path initiatives, critical CTAP initiatives, and trying to explore that. But I think we're not there yet in terms of natural history controls and to avoid placebo. I'll ask you to comment on that as well.
Okay. That is, unfortunately, that is where we are at this point. One of the things that we incorporated in the design of the study was the ability to not get natural history in these boys, but actually look at how the disease was progressing in these boys, and then be able to compare it. So they're their own internal controls. It's not natural history. And that has been very helpful in understanding the data. But we are working with a number of the consortia to move ahead analysis of natural history data, because certainly that would be an ideal situation. We're not there yet, though, as you say.
Yeah, so I know we've been, as you know, working with the consortia to think about, could we establish a protein signature? Is there a disease progression model, either CTAP, CPath, or some of the other ongoing imaging? DMD is working on this as well. So I think we're all sort of aiming toward that, but I agree we're not quite there yet. I wish we were, and someday, hopefully soon we will be, but not quite yet.
Yes.
So I thank you, Joanne. This has been really informative. We're thrilled about your progress. We're really pleased to see this edalosol and exon come forward, and I'm now getting able to pronounce it, so that's even a terrific win for me personally, who bungled it all of the time. And I do thank all of you for joining us today. We wish you all the best, and we're anxious for you to begin your phase 3, and certainly we're anxious for 2020 and hopefully an approval. So thank you all for joining us today. I hope you learned something. I learned a great deal.
And
Joanne, thank you.
Just also say that we can't thank the patients and their families enough. We understand the burden of traveling. These parents and families, they've been flying across the country, and it shows an incredible dedication that is really humbling to see how dedicated they and the investigators, the site staff, and everybody involved with the study has been. PPMD it has really been very much appreciated, and we thank everybody.
I think, Joanne, thank you for that. I think this community strength continues to humble me every day, and I just admire them so greatly. But I'm also thankful for you and for other companies to come on board and really try to really make such a difference in this disease. So thank you. Thank you all for joining us, and we'll see you again soon.